Hello, and welcome to our review on the special senses. We started our lecture on the eye. Now, I've highlighted the skin around the eye and the eyelids. The job of the eyelids and the eyelashes here are to trap things and to keep things from going in. However, the eyelids protect in other ways as well. If we sort of zoom in on the eyelids and we turn it a little bit, we're going to see these glands in here called membroidian glands. There's the ducts for the membroidian glands. The membroidian glands are going to produce a mucus that prevents evaporation. Now, there's also another membrane in there called the conjunctiva. So if I zoom in, this white of the eye is considered the sclera, but if I zoom even more in, I'm going to see, oh, wait a second, it's going up under the eyelid. If I go this way, it's going up under the eyelid. That's not the sclera, it's this membrane called the conjunctiva. That is going to produce mucus to help keep the eyes moist, to help oxygen to diffuse in. Now, there's a third structure right here called the lacrimal gland. That basically means tear gland. And it produces lacrimal fluid, which are tears. And they wash across the eye to the duct right here. And this duct is going to go into the lacrimal sac, and it goes down. So this is supposed to be the conjunctiva right there. It's a nice view of it, actually. So the outer tunic of the eye is called the fibrous tunic. It's made up of the cornea here, which is the clear part, and the sclera, which is the tough outer part. Now, unfortunately, we're not doing the cow eyeball dissection where you will spend countless hours trying to cut through the sclera. We literally destroy our scalpels and need new scalpel blades. Now, it also anchors the extrinsic muscles of the eye. There are 6 of them. There are 4 rectus muscles. The rectus muscles go straight. Now, this is the medial side, I know, because that's where the lacrimal bone would be. So this is the medial rectus. This is the inferior rectus. This is the superior rectus. This is the lateral rectus. And they move the eye in the direction that the eye is. Oops, I goofed on that one. That is the superior rectus. That class is the levitator palpebrae superioris muscle. Sneaky of them to put that on this picture. So this one goes at an angle. It's actually going to anchor into the orbit, and then there's a tendon. This is the superior oblique muscle because it goes at an angle, and it's where the trochlear nerve is attached because that's a trochleella, it's a pulley. And that is going to help counteract the rectus muscles because they tend to make you go in. And this is the inferior rectus muscle. So those are the muscles. Now, while I'm on this view, we have the optic nerve right there. And the optic nerve is going to take sensory signals out of the eye. And there's a spot in there that does not have any sensory receptors. So that's where your blind spot would be, where the optic nerve is leaving. So I have removed the fibrous tunic. And we have the vascular tunic. It has blood vessels in it. We can see the optic nerve leaving right there through it. There's the blind spot or the optic disc, and it has this structure called the choroid. The choroid's job is to keep light from scattering. So I've removed the eyebrows, and there's the conjunctiva. Bye-bye conjunctiva. Bye-bye cornea. And underneath the cornea, let's get rid of, is the iris. The iris is the colored part of the eye, and that is part of the vascular tunic. So underneath the iris is, that's part of the iris, we have the lens back there, and we have a muscle, can I find this muscle, called the ciliary body. So the muscles that muscle's not showing up, but here are the fibers that are connecting it. And the ciliary body helps keep the shape. So I've removed the choroid from this side, and we have the retina. The retina is where the receptors are. And I've taken away part of the retina, and we could see The optic disc is right there. It's going to, I have to only unhide it. It's going to be where the optic nerve leaves. There's no photoreceptors. And then lastly, this is the vitreous humor. It holds the retina on. So next we're going to talk about the ear, and the Borg queen here is going to model it for us. The outside portion of the ear is called the auricle or pinna. Its job is to filter sound in, and it goes into this hole, which is the external acoustic meatus right there. And that goes into the temporal bone. Now, there's this canal right here called the external acoustic canal, and it ends in the tympanic membrane. And sound waves basically hit here and vibrate it, and that is turned into the latest pop hit or it takes the latest pop hit and turns it into nerve impulses. Now, the way it does it are 3 tiny bones called ossicles. So, those 3 ossicles are the malleus, which is attached to the tympanic membrane, the incus, and remember on this model I told you after I made the stapes is missing. If I go here, the stapes is still missing. So the stapes should be right here. Those vibrate it and push on the structure called the oval window, and they're going to cause this perilymphinate to move. Now, the inner ear is made up of the semicircular canals, and it has the vestibule and the cochlea. Now, the semicircular canals and vestibule deal with balance. Static equilibrium is the vestibule. Dynamic equilibrium, that's movement, you're telling your head's moving, is in the semicircular canals, and may go on the vestibular nerve, which becomes the vestibular cochlear nerve. The cochlea is filled with fluid, and it deals with hearing. So we are going to look at that. So back here, I have taken most of the temporal bone off. That's what's left of it. So we have our malleus right there, our incus. There's our stapes pushing on the window, oval window. There's the round window, and it's pushing on the vestibule. Now in here, there is a fluid called perilymph. And the vibrations, listening to your latest pop song, is going to move, and it's going to hit this membrane in here at various places, and you will hear it as the latest Bruno Mars or whoever is singing currently. I've cut some of the skin off so we can see the fluid-filled sac, and it's a spiral. And I'm going to show you a cross-section of what the spiral actually looks like. So you have a continuous tube. The top part up here is the scala vestibuli, and this is the scala tympani. And this is the part where the sound waves are going to go through. They are going to hit this membrane, the vestibular membrane, and cause vibrations. That moves the endolymph in the cochlea, in the organ of Coti, and that pushes on our hair cells right there. Now, the sound then goes back out the scala tympani. So your tongue has all these raised papillae. These are my favorite. Those are the valate ones. But you have various papillae. And on some of the papillae, you have taste buds. Taste buds are bundles of gustatory cells. Those are cells that taste a specific chemical. And it takes the information to the brain by either the facial nerve for the anterior 2/3 or the glossopharyngeal nerve by the posterior third. So these are the taste buds, and contrary to popular belief, all the taste buds can taste all the tastes because you have all the different cells. That thing you learned in 5th grade, it's from an incorrectly done experiment that's almost 100 years ago. Lastly, we have olfaction. Here's the olfactory bulb. And the olfactory bulb is going to have neurons that go through the cerebriform plate into the nasal epithelium. And these bipolar cells have these cilia on them. They're actually dendrites. And they have receptors for different chemicals. And you smell vanilla, it'll stimulate that one, and that one. You smell skunk, it'll stimulate that one, and that one. And that takes the odors to your brain by the olfactory nerve. So smell is olfaction. Vision is optic. We have glossopharyngeal and facial for taste. Vestibulocochlear for hearing and balance. And I didn't have the retinal layers on that other one. The retinal layers are right here. We have rods which are these rod-shaped ones, and cones, which are these cone-shaped ones, that are photoreceptors, but they face backwards. So this is the choroid. That is actually the back of the eye. So there's the choroid right there. And these are going to detect light, but the light has to go through, bounce off the back, and stimulate them. Cones detect color. Rods detect black and white. Now, rods tend to converge on the second cell layer, the bipolar cell layer. The bipolar cells that are connected to the rods tend to converge on the ganglia cells. Now, this is part of the central nervous system. It was just poorly understood, so they called them ganglia cells. Now, each cone gets its own bipolar cell, which gets its own ganglia cell. So color vision's a lot more accurate. So you need to know the layers. We have bipolar cells, we have rod and cone layer, and the ganglia cell layer. So that has been our 11-minute review of the special senses. Thank you.